have Dr. Maltecca presenting for us. Christian Maltecca is a professor of quantitative genetics and breeding in the Department of Animal Science at North Carolina State University, United States. His research program focused on genomic improvement in livestock, the optimization of statistical methods and breeding schemes under genomic selection, and the effectiveness utilization of intermediate omics in selection programs with the use of parametric, non-parametric methods, and machine learning methods. So I will turn it over to you now, Dr. Maltecca, and can get it started.
Thank you. Hope you can hear me fine, and thank you for the kind invitation. This is the title of the presentation, and it's a real pleasure to give it to you. I have a lot of collaborations with the people at CGIL, so you see that some of them will be in the title of the presentation. And I apologize for those of you who were at the ADSA meeting this summer. This is relatively close to that talk because it was closing time, and I am presenting about inbreeding and genetic diversity. So let's dive into it if we can. Okay, so what I'm going to do, what I'm going to talk about today is a little bit of a background on why we care about managing genetic diversity, and a little bit of what happened, and a little bit of what my research is in this area. Some of these, especially in the beginning, some of the things that I'm saying are relatively general. Some of the work that I will be presenting is more specific to dairy, because in dairy, managing diversity and especially managing inbreeding is a problem that is a little bit more present than in a system like pig or poultry, where the entire control of the production system is in one or few hands. So typically the management of variability is a little bit easier to constrain. In dairy, because of the very horizontal structure of the dairy breeding process, it becomes a little bit more complex. And that's what we're going to focus on for the rest of the presentation. So what is managing the diversity? And you have this horrible picture here where you have basically in the middle, you have standing variation, which is what we can work with, which is basically what is available to us to do selection, and that's what we normally do. How do we deploy? We're not going to talk too much about how we deploy variation, although this is another interesting topic and something that could be covered perhaps in another talk, not by me. So we have two main areas, right? On the left side, we have the genetic load, which is one of the concerns that we have when we do selection. Selection is basically a process of inbreeding a population, and with that come a couple of unwanted consequences. The first one is the accumulation of inbreeding load, so the accumulation of homozygous recessive, in this case, a homozygous state in the population. The second one is the mutation load, like the creation of new mutation that might or might not be favorable. Some of them might be favorable for selection. The one we're interested in controlling are the ones that are not. So in this case, we work with selection and purging, but actually if I have to basically go through what are we concerned with when we talk about genetic load, is like we're all going to die, right? So individuals are losing fitness, individuals are not going to be able to sustain production or just remain in the herd long enough to repay for themselves. On the other hand, and this is typically more of a short-term concerns, like the accumulation of inbreeding concerns the population today, and then there is the usual long-term concern, which is related to how much variation there is to work with, and this is typically due to the selection process, the selection limits. We actually don't know much about selection limits. It's typically due to the architecture of the traits, the genetic architecture of the trait. It is also due to the operational model, which is the way in which we model genetics, which is typically just an approximation of the unknown biological process that drives the genetic architecture of the trait under selection. And typically the concern in this case is there's nothing going to be left to select, right? Clearly, I wanted to put these two large sort of we are all going to die and there is nothing left to select is sort of an exaggeration, right? So we're working within these two large frames, which are not truly going to happen either in the short term or not if we manage population, or even at the rate by which we are managing population now. But they're the two biggest fears that we have to work with in dairy selection. So we'll start with the genomic load, with the genetic load. And I'm going to talk a little bit about why we worry about and what are some of the constraints that we have when we're working with the load, with the genetic load. We typically are concerned about homozygosity or some measure derived from homozygosity, identity by descent, typically in using pedigree information or SNP information if we're using genomic information, and we typically resort to genome-wide homozygosity, right? The reason for that is that it's easy to obtain, but the drawback of that is that genome-wide homozygosity, however we measure it, is a bad indicator of the genomic load, right? We use it because it's identifying deleterious variation, whether it is lethal or sublethal. Here I have sublethal because typically lethal is a little bit easier to identify. Deleterious variants are relatively difficult because the genetic architecture is also a direct function of the selection process. We tend to think as the genetic architecture as something fixed, but we are shaping a population as we select. And typically, because inbreeding load, we're not going to talk about mutation load because we don't know much about the mutation load, especially in cattle under selection. But we don't know when we talk about the inbreeding load, we typically still don't know how much directional dominance there is in dairy cattle, because directional dominance is relatively difficult to map. It is difficult to map additive variation. Dominance variation is a little bit more complex. So the other big thing that we often forget when we are talking about some of these things is that we're assuming that our population are not under direct selection for fitness. So typically when you have deleterious variants, those tend to be eliminated in a population. Natural selection normally does that trick, but we are sort of subjecting our population to sort of very intense directional selection, even for fitness, right? Even if you're just talking about the culling exercise within the farm. But one of the issues with that is we truly don't know how much that is happening or not. There have not been a lot of studies that have looked at how much the selection that we're doing we are actually is actually purging deleterious variants in the global herd. Okay. Also, because typically we're not very good at measuring fitness, right? We typically in either in research setting, but also in selection setting apply to a breeding population. We do not define fitness in a very coherent way. We use proxies, we use something that we think is related to fitness, but might be fitness under certain condition and might not be under certain different condition. So in the end, if I have to put everything together, to understand the genetic log, we have to sort of understanding what is the architecture of inbreeding and the architecture of inbreeding depression. But this is very complicated to do in breeding population. If we were working with Drosophila, typically we would do a nice, very well designed balanced experiment, a selection experiment, but this is not economically feasible. The biology, especially in the area of the organism we're working with, prevents us from doing selection experiment, and typically those were done in the past. In the 50s and 60s, but there was not a lot of incentives back then to continue them, and now those typically have been phased out, and now it's probably too late to start some new selection experiment. And simply this sort of question, how much is the inbreeding load, how much is the mutation load, how much selection and purging are acting on these things, are very hard to answer retrospectively by just looking at a cross-section of the population that we have right now in the breeding, in the global breeding population. So understanding the nature of genetic variation in breeding depression is pretty hard. So what can we do? Well, we can increase the focus on deep phenotyping. We now have a lot more possibilities. We just had up until few years ago to measure things that are related to the fitness of an individual. We have camera, we have sensors, and those will become more and more popular as time goes by. Functional annotation has been lacking for a long time, especially in livestock populations, but there has been a large push in the last few years of in increasing the amount of information that is available, and how do we increase integration into this functional annotation in models that are either to make inference about the nature of inbreeding depression, or even just predicting the function of inbreeding depression becomes a large research topic. Of course, increase the number of genotyped individuals, but this is typically not a problem for dairy population where we have millions and millions of individuals that are genotyped. The omnipresent refined analysis tool, integrate model organism approaches. We need to look a little bit further out than what we typically do. Investigate the potential of advanced technology, which is typically the case, for example, of gene editing or some other reproductive technology that can help us sort of hijack the process of understanding how inbreeding is accumulated, and of course data sharing, right? So whenever there are different populations working on the same project, on the same problem in this case, it's always useful to do that. So in the short term, though, managing homozygosity, I shouldn't say managing homozygosity, but managing the accumulation of homozygosity remains sort of a logistic project that is better managed at the population level, at the global herd level, some sort of with the use of some sort of this genomic-wide homozygosity measure. So the second one is that perhaps it's equally important, has consequences on the long term, and it's how much can we select at the rate we're selecting now before we hit a wall, right? So we don't know. And the truth is we don't know, no one knows, but typically there are a few things that we can learn from looking outside of what we have seen. The picture on the right here pictures the Illinois corn oil experiment, which is an experiment that has been going on for more than a century now, and those are selected lines for oil content in corn, in maize. And so you see now we are this stops at 216, but the experiment is still going on. We are at more than 120 generations now, and still the selection, of course, the selection for the diminished corn oil reached a plateau when the corn oil was absent. But for the remaining one, we're still selecting, and we're still making progress. So based on what we assume, typically with a very simplistic models, we should have run out of variation to work with a long time ago. If you say maize is probably too far on a genetic architecture from what we're used to, and you want to get a little bit closer, there are plenty examples. This is an example in poultry, but there are plenty examples where you see that even after 50 generation, where we would expect that the variation available to do selection was gone, we're still able to make progress. No one really knows exactly why, the truth, but there are several theories. Probably the easiest one to adhere to is that there is conversion, right? I talked before about operational models. I talked before about operational models, which are basically the easy way for us to statistically describe the reality, but in reality, things get more complicated. There is conversion of epistatic effects or dominance effects into additive effects as time progresses, and so we're able to recruit them, so new variation is recruited. So this is not a problem of is there enough variation? Perhaps we should start asking ourselves, are we able to select fast enough if we are changing our breeding goals? For example, if we need to select individuals that are, for which the fitness is completely different under different scenarios, let's say, increased heat waves, can we do it fast enough that the population is not suffering from the point of view of the overall fitness? So these to give a general idea of what I think the problem is, or how I think about the problem. And here I'm going to present very few results. Of course, I'm not gonna try to present everything about all the possible things that I've just mentioned now, right? So if I still stay in the field of looking at how much variation there is and how I can keep selecting and talking about epistasis and variance conversion, there are a few studies that are coming out that are looking for epistasis, and this is one of them. I think one of the most recent one, if nothing else. I'm putting it here because it's in U.S. Holstein cattle, and there is quite a bit of signal for epistasis in dairy. As I said before, often the operational models that we use are very ill conceived to detect higher order interactions, and higher order interaction typically are poorly captured by any highly parametric model, but still there is evidence that are out there. And even in simulation, you can see that there is this variance conversion and things that are coming from both dominance and epistasis tend to be recruited by selection, so that actually the response to selection gets better over time, or keep going over time, even when additive variation might not be replaced by other additive variation, and it's replaced by something that is converted to additive variation. So typically in the past, this is another little aspect that I think it's important, and it's related to the epistasis. In the past, it has been proposed to use recombination variation. There is a genetic component for recombination in dairy. This has been proposed as a way to maintain variability and using gametic diversity as a selection goal for dairy. But based on what we said before, or what I presented before, based also on the fact that these models typically tend to overlook the complexity of how variance is transformed, here's just a little bit of an oddball. Not always, especially if there is epistasis, not always creating new haplotypes is advantageous. And actually, there is a few papers that show that that actually decreasing recombination for selection for genetic architecture by which genetic variants have been co-selected, so that sort of we fail into the additive infinitesimal model, might be better for the response to selection than actually increasing the recombination. So this is just to say that it's a little bit more complicated than what we normally assume. So, and now I'm going to dumb it down by telling you a little bit of what we are doing, right? What we're doing with my group, and in collaboration with people at Guelph and CGIL. So I'm going to talk a little bit about what can we do to understand inbreeding, considering also the limitation that I talked to you about before. And so I'm going to talk a little bit about how can we use functional annotation to dissect inbreeding. I want to talk a little bit about the heterogeneity of inbreeding and environmental load, because we always assume that inbreeding is a sort of fixed, inbreeding depression is a sort of fixed quantity that pertains to or fixed property that pertains to a population. But in reality, there is this whole interplay between the challenge of an individual and the possibility of expressing inbreeding depression. And of course, because understanding inbreeding has consequences on cow fitness, a little bit of the effort that we're trying to do to make sure that we sort of try to understand cow fitness a little bit better. Very little about managing genetic diversity. So it's basically, I'm going to only talk about what is the impact. Typically, we manage the population, the global population, by by sort of curtailing the future inbreeding, especially in dairy, especially in North America. In some other countries, they might be using something closer to optimal contribution selection, but typically here in the United States or in Canada, we tend to manage the population by looking ahead and preventing the future generation to accrue too much inbreeding or too fast. And finally, one of the things that I'm going to talk about in just a little bit, I put it there as a bonus, we now have a little bit more of leeway into using more complex models that are not model-driven, that are not as parametric as we used to think in terms of quantitative genetics. And I'm only going to hint about large language model as a way to identify deleterious variation without a predefined experimental design to look at things. So I'm going to talk a little bit of functional annotation to dissect inbreeding. This is work that we have been doing in collaboration with CCB, the University of Florence, and University of Maryland. This was under review the last time I presented, but now it's published. You can find it on Journal of Dairy Science. And the idea here was to look at functional annotation obtained through sequence information, and how can we use that to sort of separate inbreeding depression based on classes of functional annotation. This is not very dissimilar to what you would expect or what you have seen done from us and from other people in trying to categorize inbreeding based on the length of inbreeding with ROH or something similar. In this case, we have done something a little bit more refined or a little bit different by looking at where a variant fell in terms of its functional annotation. So we use 5 functional annotations. So the coding region, the promoter, the untranslated region, the introns, and those are the GURPS, which are highly conserved regions that tend to have biological significance. So we use Jersey because Jersey are a population that is typically has accumulated a little bit more inbreeding. And we had about 250 Jersey cattle for which we mapped sequence information for about 10 million SNPs. And we actually did find, so we actually calculated inbreeding depression by classes of annotation. We also looked at other things here, for example, whether the availability of a more or less dense marker panel does impact the amount of information that you can tease out. But just to summarize the results, there is basically an enrichment. This is on the y-axis, you have the enrichment ratio, which is basically telling you how much more likely it is a variance of that class to impact inbreeding depression compared to the average variant. And you can see that in all cases and for all traits, in this case the traits were milk, fat, and protein, variants that were belonging to the untranslated region and the GURP, which are what we would expect typically to be enriched in inbreeding depression, were indeed enriched, and they were across traits. We have some other data that I'm not going to show today, for which this is confirmed also with other kind of analysis and heritability partition, but the idea here is that actually having functional information does provide an extra layer of information that allow us to sort of tease out better what the effect of inbreeding depression is, right? Some genome annotation have a disproportionate effect on inbreeding depression, and these might be more vulnerable to deleterious mutation. It means that if you're accumulating inbreeding depression there, you might have a higher chance to accumulate something that is deleterious by nature. So we can use it to disentangle a little bit more inbreeding and inbreeding depression. And here, as an example, you have 2 individuals, 2 Jersey cows, for which that they have more or less the same level of inbreeding, but different accumulation of inbreeding in terms of classes, right? So we can tease out these 2 individuals that have more or less the same inbreeding by characterizing them a little bit better. It's no different than doing it, for example, for length of ROH classes, but it's a little bit more, a sieve that is a little bit more finer. So the other thing that I'm going to present today is like a little bit of the heterogeneity of inbreeding and environmental load, what I defined before. What the question that we were asking is, what is the effect of exposure, of challenging, that is environmental challenge. And in this case, we use heat stress, and this work was done particularly by Francesco Tiezzi and by Joao, who was a postdoc that worked with us and with data that were from the Italian Holstein Association and also in collaboration with the University of Parma. So the idea here was to look at inbreeding depression, but look at it for classes of environmental exposure. So what we did, we had information on test day record on a sizable number of cows. For this, we were able to calculate THI, but also to obtain relative humidity and temperature. And we made sure that all the cows had equal exposure to classes of environmental exposure. So we divided up THI or the other measure in from the most comfortable to the least comfortable. Sorry. And the first thing we looked at, well, we wanted to check whether at increasing this comfortably condition, what was the production. So, and as you would expect, the production decreased. In this case, it's milk yield for relative humidity. And you see that it stays a little bit, it decreased up to a certain level, and then it truly started decreasing. But then we also wanted to see, okay, is inbreeding depression the same across the different classes, or is there a F by E interaction? And needless to say, you have this like sort of U shape where at the most comfortable condition, on the left you would have cold stress, on the right you would have heat stress. At the most comfortable condition, of course, the inbreeding depression is a little bit less, whether you measure it by pedigree or genomic information. As you get farther away from each of these classes, it becomes a little bit more prominent. I should say this was a poster when it was submitted, but I think this was accepted 2 days ago, so you should see it coming out. No, sorry, that's another one. This was submitted, but it's still under review. But again, another interesting thing is that if you look, you can quantify. In this case, we looked at we did a little bit of data wrangling, and we look at the 5 most inbred and the 5% most inbred and the 5% least inbred individuals in the population, and we look at the average inbreeding and the average inbreeding depression. You can see that there is clearly an indication on F by E, right? This is the clear. If this was a G by E, you would say, okay, this is a G by E. As the environment changes, the genetic effect is different. The same sort of speculation you can make it for the inbreeding effect, which if you look at the mean, which is normally what we use to calculate inbreeding depression and to use penalties for expected future inbreeding or for mating design or stuff like that, you see that individuals that are the most inbred are actually probably underestimating inbreeding depression, especially in challenging conditions for some of these individuals. So some of the things to look at. Even if we consider inbreeding without considering F by E and the environmental challenge, can we look at it in cow fitness? And this was the paper that was just accepted from the same collaboration from Joao again. And in this case, we looked at stayability. The reasons for this, we started looking at this kind of data on beef cattle, and stayability was something that is typically more close to what beef cattle breeder think about. But we thought it was a good idea to look at it in Holstein cow too, because it's the probability of making it to the next lactation, right? So it's a little bit different than productive life. That is more what we're used to see, especially in North America. And again, here we use stayability from 1 to first, second lactation, second to third, third to fourth, and so on and so forth. We use 2 data sets. That is why you see 2 lines here. The first data set was only including individuals for which we had genomic information, and the second one was for individuals for which we had the whole population. We wanted to make sure that we didn't include any bias due to the fact that typically we do not genotype all the individuals, but we tend to reserve genotyping for individuals that have a slightly better genetic merit. And in both cases, you can say that for stability, as inbreeding gets higher, the probability of making it to the next lactation gets smaller, right? So perhaps this is not particularly earth-shattering. We would expect something like that, but I think it's interesting to see that it's not a small effect. It's a relatively large effect. At least it is a relatively large effect in the Italian population. We are now replicating some of these exercises in the Jersey population that you have seen before. And again, if we look at the usual trick of looking at old inbreeding versus new inbreeding by looking at ROH length, again, you see that inbreeding that is long and that has not had the chance to be purged from the population is getting a little bit more depressing than the other one, right? Ideally, for the ones that have very small, you have that something that you typically very small ROHs, you have the typical effects either negative, it's either null or slightly positive. It means that it has been recruited, it's under selection, and we're not cleaning it perfectly using the additive effects. So what's more interesting is that if we, in this case, we looked at the predictive probability of remaining in the herd, so to making it, at each lactation, and we look at the genetic trend and the average phenotypic trend, and you see that the genetic trend has gone up in the previous generation, would go up in the previous generation, but the phenotypic trend is going down. And short from saying that this is actually all due to inbreeding, there is maybe a case to make that inbreeding depression might be starting to get to the point where it's getting in the way of the additive accumulation, or the accumulation of favorable variants. So it's very so now we talked a little bit about the left side of the picture, right? The genetic load and how to understand inbreeding depression. Now I'm going to switch for a second to the right part of the equation and see how do we manage the long-term variability and also the inbreeding in the population. Here I'm going to present just a small simulation that we are doing, and this currently drafting the paper, where it's going to be submitted, I hope, soon. This was the result of something else that we were doing. A student, Emmanuel Losada Soto, was working on some strategies to do germ plants exchanges to manage population diversity over the long term. This was published last year in Journal of Dairy Science. But the structure of the simulation was there, and there were a few questions that were not answered when we started looking at them. So we thought, okay, what if we started looking at what happened when we penalize future inbreeding? What happened to the genetic architecture? What happened whether we use pedigree future inbreeding, genetic future inbreeding, or runs of ROH future inbreeding, and what happened if we assign different penalties? So what happened, typically what we wanted to track was the additive variation, which is typically what we have done in the previous simulation, but we were also interested in saying, okay, which one of these strategies is managing the dominance genetic variation, which is something that is typically less seen in this population, and also what happened to inbreeding depression, what happened to the accumulation of inbreeding depression. So I'm gonna only to present, we have several scenarios. I'm gonna only to present one where the dominance variation was fixed at 9%. Don't ask me why 9%. It seemed like a value that made sense when I started simulating the data. On the left side here, you have the accumulation of genetic homozygosity. And as you can see, there are different scenarios for which we apply the penalty to the selection process, just like in a expected future inbreeding. And we look at the accumulation of homozygosity, and of course we always have a positive control, which is using the true breeding value, and a negative control, which is random selection. As you would expect, if I use the true breeding value, but even if I do not penalize anything, things get sort of out of control, or the homozygosity accumulates a little bit faster. The only scenarios for which the homozygosity is reversed, so they are actually increasing heterozygosity, are the ones where you apply penalty, whether you apply the typical genomic relationship penalty or you use ROHs. The point of using ROHs is that you have a little bit more freedom to put selective pressure not on the genome, because you're only partially penalizing inbreeding. You're penalizing inbreeding only on certain regions which have accumulated long stretches of homozygosity. And of course, when you look and what happened when you do that, if you penalize inbreeding based on ROH, typically you obtain a slightly better response in selection than if you do it with GFI, simply because you're a little bit more liberal in penalizing inbreeding. The opposite is true when you look at maintaining dominance variance, because if you do penalize inbreeding based on the overall genomic inbreeding, then you're basically promoting heterozygosity a little bit more efficiently, so that typically the scenarios that use the GFI rather than the ROH-FI tends to maintain more genomic variance. But in general, actually penalizing inbreeding is a good effort, also to maintain dominance variance in the population, which is sort of something we typically tend to overlook. In all cases, inbreeding depression tends to accumulate, and although I wouldn't put too much thought into what this means, because this is probably the results of how we simulate the data more than exactly what's going on at the biological level. So the point here is that penalizing future inbreeding is effective at constraining the accumulation inbreeding in the population, and both GFI and ROH-IFI do a good job in doing it. You get a slightly better response to selection if you penalize ROH on the medium term, at least. You do maintain a little bit more of dominance variance in the population if you do penalize based on GFI. Because dominance variance tends to be recaptured by additive variation as time passes, the interplay between the two might be a wash, but further simulation will be needed for doing that. So the last thing I'm gonna talk to you about today is going back a little bit in trying to understand the architecture of deleterious variation and inbreeding depression. I'm going to put here one of the things that I think, one of the best studies that I think has been published recently, at least in dairy, in trying to understand deleterious variants. And again, there has been a lot of success in identifying deleterious variation that are either lethal or near lethal. And that's simply because we have a very good mechanism to do it. It's the absence of homozygous in the population. It's very easy testable. You can use a chi-square to do it, and then you can do something else to look at these things and confirm it through molecular means or to more sophisticated analysis. But for partial recessive or recessives that have like very small effect, but cumulative effect might be large due to the accumulation of inbreeding, there is a little bit less that has been done, and it's a little bit more complicated because of what I said before. So this, which was published 2 years ago, one and a half years ago, has a very comprehensive way to look at the distribution and the mapping of partial deleterious variants. And I think it's something that in US, at least in North America, we're sort of lacking behind. And I think I would like to see some of these exercise done more in our population. So if you haven't read it, I encourage you to look because I think it's a very well done study. One of the things that I want to point out with this is that this is sort of a collective effort, right? So to go through the whole, I'm not going to the whole experiment, to go through the whole thing from A to Z requires a little bit of integration of different players. And here you have a figure from a paper that was published this year from a colleague of mine, John Cole, which actually highlights how the structure of the dairy population and the dairy industry in North America, it's a little bit more fragmented, and there are several actors that are sort of intermingling in collecting deleterious variants, whether those are monogenic genetic defects or polygenic genetic defects. And this makes it very difficult to design experiments or to do analysis that are conducive to understanding inbreeding depression. So he proposed a new system to do that. I'm not going to go into that, but I think it's a very good idea. I think in Canada, Christine Bays has proposed something similar that is currently under investigation or is under implementation. And I think this is one of the keys when we go back into data sharing, creating new and larger data set to understand inbreeding depression. It's something that is particularly important. But in the meantime, is there anything we can do or we can do associated with some of the things that we're doing with some surveillance system to basically prevent deleterious variation to start and accumulate in a population? And here is an idea that we had. There are now a lot of large language genetic models. If you're familiar with ChatGPT, Gemini, or whatever it is that you use to fix your code, like I do, those are very, very powerful models, and they are foundational models that are large language models that have very nice properties. The same models can be applied to genetic variation. And the idea here is, can we use these models that are sort of context dependent but not model dependent to predict deleterious variants in bull based on large language model? And the premise of this model, let me see, the premise of this model are that if you train this model on a very large population that does not even need to be a dairy population or a livestock model organism population, but can include all of this, right? So this is basically what is normally called transfer learning. You can actually sort of predict what you expect a single variant or a new variant, if you have a new mutation, to be in terms of being possibly deleterious or not. There are a few things, I don't know if I want to go too much into this, but the idea here is that you using the large language model is not particularly complicated in terms of conceptually what they do. What they do is they chunk up a sequence, whether it's a sequence of word, a sequence of images, or a sequence of anything, and they create tokens. And tokens are nothing else than the split of a sentence in this case, like the cat sat on the mat, or some genomic sequence, right? So those are basically tokens. They don't have any meaning. But once you have tokenized a sequence, you can actually embed it. And embedding is like what makes these models particularly useful, because they're converting tokens in numerical vectors. And whenever you have a numerical vectors, then you can apply some sort of algebra to it, right? And how do they use this model, this algebra? They actually use it to capture some semantic relationship between things, right? So similar tokens have similar embeddings. Tokens that are next to each other tends to behave the same way if they have some sort of semantic relationship. And then what makes it easier is that it allows to use any of these machine learning algorithms to be applied, right? Because those are numerical values, numerical vectors like any other that we're used to work. And they can work for text as they can work for DNA bases, right? So basically, the idea here, can we use next token prediction? Can we predict what the next piece there should look like, or what is the semantic meaning of that particular one, right? So, and that's basically the whole trick. One of the interesting things is that you have that attention mechanism that actually allows the model to learn from contiguous information in an efficient way, so that it is able to sort of prioritize what's important and what's not. So this is typically how they work. So one of the things that my colleague Jisai Jiang here did was to look, can we see if we can predict whether a variant is deleterious or not, just purely based on its sequence, so without knowing anything. And here we cheated a little bit because we needed to know the ground truth. So we went to OMIA, and we got single inherited deleterious variants for different animals, and we put them all in a data set. We masked them, and we basically did a prediction exercise, right? So we took some control variants from the sequence, and we said, okay, we anonymized them, and we use Evo2, which is probably the state-of-the-art model to do this kind of exercises. So we didn't invent the model, we retrained on a model that already existed. And then we said, okay, can we predict whether that variant, without knowing anything, without the model knowing anything about it, but with us knowing the ground truth, because it's annotated in this case, can we have the model predict whether that variance is a disease or not? I'm going to make it very short for you, but it works, okay? And it works really well if you use random variants. So if you don't choose them with any particular meaning, it works a little bit worse, but still very good if you use variants that are only on the same class of the variants that you're trying to predict. This is to make it a little bit more difficult, because in the first case, it's just basically trying to separate micro categories, while in the second case, it's trying to separate variance context within category. So it's a little bit more tricky to do. So I will not say too much about this, but it works pretty well. And now the paper is under the paper is under revision. So we hopefully will be out in the next few months. But so here is a potential work how using some of these methodologies can be integrated into the real world, right? So to say you have a new bull, the new bull is born, you sequence the bull, you want to know whether that bull should enter the breeding, the global herd, and be put into production and produce semen. What I would like to see is that you apply Evo2 or any of these large language model, and you produce some sort of a risk score of all the new variation that it carries, new mutation that it carries, and you categorize it either in high risk, the variants that the collective number of deleterious variants that he carries are probably a risk for the population, or it's a low risk. If it's a low risk, then you calculate future inbreeding as you did before, and you can use functional notation or any other more or less sophisticated way to use global homozygosity to manage his risk in the whole population, and you put it in the breeding pool, and then it goes back to reporting, to some sort of reporting exercise. If it's at high risk, you do a value system. How good is it? Okay, so it's very good. Then you can go back, and if you have the luck of having, or in the future being able to apply some of these advanced technology like gene edit, you can think of gene editing that individual and re-putting it back in the breeding pool, or if you can't, you just call it. Okay, and that provides also information for next bulls that will come around. So here you see how you can actually manage a little bit of everything, because you do have management of the overall genetic variation of the population with future inbreeding. You can use a little bit more refined or a little bit less refined, but you also have sort of a management of the genomic load of individuals. And this clearly is the first attempt. There can be many others that you can use, but I think that's one self-contained way in which we can manage genetic population. There are other applications clearly that are related to the use of large language model. This is only one of them. If you're interested, hopefully the paper will be out soon. So to conclude, before I put you all to sleep, I think collectively over the last 10 to 20 years, we develop a lot of better tools to understand inbreeding and inbreeding depression. We do a better system we need to do more research. And with this, I just want to tell people that work with me on this, all the collaborators, and the other two, which Christine and John Cole, were largely the one with which I share ideas and collaborate. And that would be it. Thank you.
Thank you very much, Dr. Maltecca, for your presentation. I'd like now to open the floor to any questions, either from the room here or from those joining us online. We do have like time for 2 short, quick questions. Anyone online or in the room?
Yeah. Can you
go ahead?
Hi, Christian. This is Lucas here in the room. My question was more just out of curiosity, if you had looked into the return, like the financial return of keeping cows that are inbred because they're already producing more versus trying to correct for that as is shown in the models, and seeing what's the return of that, because I would assume that the less inbred cows would probably produce a little bit less. So what's the trade-off between those two options that you would have?
So can you hear me? Yes, I don't have the plots here, but there is always the trade-off between maintaining a cow that is less inbred in your population and paying a penalty. I think the cow is a little bit less of a concern to me than the sire is. I'm more concerned in terms of genetic accumulation of inbreeding. I'm more concerned about the overall population and the contribution of the sire. At the herd level, I haven't talked about any of the mating programs. Inbreeding can be managed as a mating issue, so I'm less concerned about that.
Thank you.
Any other questions online or in the room? I do have one question. Does inclusion of resilience-related traits in genomic breeding values models help maintain diversity maintain diversity better than selection on production traits alone?
So we don't know. So to the extent that anytime you include any new trait in the index, the selective pressure on any of the other traits becomes lower, and then possibly you maintain more genetic diversity because you have to increase the pool of genes that you're including. Yes, in that sense, yes. I don't know if directly selecting for resilience sort of targets more dominance and maintaining of heterozygosity than the other ones. I'm not sure.
Thank you. Any other question? No? Oh, thank you so much, Dr. Maltecca. This has been a great discussion. So due to our time, we're going to finish here. And thank you, everyone, for coming. The recording will be posted on YouTube, and I will share more details about that with you later, Dr. Maltecca. See you all next week. Have a nice weekend.
Bye now.
Bye-bye.